Hello, good evening to one and all of you. From today on, I'll be taking UG teaching classes also in the Pathway channel to augment the classes by the PGs as well as the budding pathologists. And then for the UG students also, it can be very helpful. So today our topic is Introduction to Kidney Histology. So before coming to the histology proper, you should know certain anatomy of the kidney. So, kidney is a pair of bean-shaped organs present in our body, and they weigh approximately 150 gram. Cut surface, it has three main structures, that is the cortex, the outermost part, and then you can see this triangular areas. They are the renal medulla, and then there is renal pelvis. Okay, so this part is renal pelvis. And then you have the vasculature which has renal vein, renal artery, and then you have the ureter that collects the urine and goes through this to the urinary bladder. So what is the function of the kidney is mainly to filter blood of the impurities and then reabsorb the essential molecules before it is excreted through the urine, and then it secretes the waste products which was excreted through the urine. So its main function is to filter blood, okay, to take away the impurities as well as reabsorb certain essential molecules. So what is nephron? Nephron is the functional unit of kidney, okay? And nephron has components like glomerulus, Bowman's capsule, proximal convoluted tubule, distal convoluted tubule, and loop of Henle. The function of glomerulus is the main filtration part of the blood, and then tubules that come after the glomerulus handles mainly the reabsorption and the secretion. There are two types of nephrons in our body. They are cortical nephrons, which are most common, and the juxtamedullary nephrons that constitute the 15% of the nephrons. And the juxtamedullary are more important for concentration because they are long and they tubules are mainly to handle reabsorption and secretion. So as you can see here, this is a classical nephron picture. So which shows glomerulus, and then there comes the proximal convoluted tubule, then there is thin descending limb of loop of Henle, and then there is thick ascending limb of loop of Henle. This is the loop of Henle which looks like a hairpin loop, and then there is distal convoluted tubule, and then the collected urine goes through the collecting duct, okay, to the renal pelvis, and the glomerulus is covered by Bowman's capsule. So there are two types of nephrons. I said cortical nephrons are the nephrons that reach only till the outer medullae, and the juxtamedullary nephrons are little more long and their loop of Henle reaches till the inner medullae. So they are most important for concentration of the urine, and then the rest both the functions are same, the filtration of the blood. Okay, so you can see there is glomerulus PCT, loop of Henle DCT, and then every glomerulus DCT gets converged into a collecting duct that will release it in the urine into the renal pelvis. So I am just showing it. See, basically you have renal medullae which has renal pyramids, okay, and then the pyramid tip is where the collecting ducts converge. So there is cortical nephrons, and then there is juxtamedullary nephrons also. So the glomerulus is present mainly in the cortex, and all the loop of Henle comes into the medullae mainly. PCT and DCT is also mainly present in the cortex. And then the collecting duct enters through the medullae mainly, and then it releases into the renal pelvis. So what is glomerulus? It is a functional part of the nephron that filters the blood mainly. Okay. So the blood is filtered in the glomerulus, and glomerulus contains capillary loops, okay, that are formed by blood vessels, and they are supported by mesangial cells and they are surrounded by podocytes. And the filtration membrane is formed by three layers, that is the capillary endothelium, basement membrane of the capillary, and the food process of the podocyte. I'll show the figure later, it will become much more clear. Then the selective barrier, that is the filtration barrier, can filter molecules by size up to 70 kilodalton only. So below that, those molecules are not filtered. Means above that, those molecules are not filtered. That is more than 70 kilodaltons. Most of the proteins come under that category. So most of the proteins are not excreted through the urine. Okay. And then the filtration also depends on the charge of the molecule. So coming to the ultrastructure of the glomerulus, if this is the glomerulus which contains afferent arteriole and the efferent arteriole. The renal artery that comes, it forms a loop like glomerulus and it goes off. So which enters the glomerulus is called afferent, which exits the glomerulus is called efferent arteriole. And the capillary loop that is formed, if you take a cross section like this, okay, you know glomerulus is covered by Bowman's capsule. So the Bowman's capsule has two layers, that is parietal epithelium and the visceral epithelium. The parietal epithelium of the Bowman's capsule is what you see in this yellow color. Okay, and then visceral epithelium of the Bowman's capsule is the one which you see in blue color, which forms like food processes that are called podocytes. Okay, so this visceral epithelium is formed by this blue color cells and it has podocytes. Okay, and then that is Bowman's capsule. And the space between this visceral and the parietal epithelium is called the filtration space or the urinary space present in the glomerulus. Okay, inside the capsule you can see capillary loops. You know this is glomerulus is formed by capillary loops. So these capillaries are these cut sections which contains RBCs. Capillaries are blood vessels only, so they contain RBCs. And then they have some supporting cells called mesangial cells also that you can see in the screen. So this figure is very important to understand kidney pathology, especially glomerular pathology. So you have Bowman's capsule, which forms parietal epithelium, and visceral epithelium that is formed by podocytes also. And then you have capillary loops, and that are supported by the mesangial cells. Now, filtration barrier, how it happens is that the capillary loops, blood enters through the capillary loop, and then will enter and it will go through the efferent arteriole also. So while passing through these capillary loops, the barrier, that is filtration barrier that you can see that is running like this. Okay, so the blood gets filtered through this filtration barrier which contains three layers, that is the vascular endothelium. Then there is this pink color you can see in between these blue processes, that is the basement membrane of the capillary, and then you have the visceral epithelium of the Bowman's capsule that has the podocytes. So the filtration barrier has three components, that is vascular endothelium, the vascular basement membrane, and then the podocytes or food processes of the visceral epithelium of the Bowman's capsule. So the blood comes through the capillary and gets filtered, and then the urine that is formed in the urinary space between the visceral and parietal epithelium gets passed on from the urinary space to the PCT, loop of Henle, DCT, collecting duct, and to the renal pelvis. So if you see in a 3D electron microscopic view, this is how exactly a glomerulus looks. See how beautiful it is in the ultrastructural electron microscopic view. These colors are not real, they are just projected by the AI. And if you go more microscopically in electron microscopy, you can see these are the view from the visceral epithelium when you remove the parietal epithelium. Okay, in visceral epithelium, you can see these are the visceral epithelial cells that process and give podocytes that you can see here beautifully. like tree branches. So these are podocytes, and inside that violet color you are seeing is vascular basement membrane. Inside that you will get the capillary endothelium. So these three layers form the filtration barrier. So from inside these capillaries, the capillaries which are inside, from their blood will filter through this barrier and will come to the urinary space here. So next, after from the glomerulus, the urine passes to the PCT, that is proximal convoluted tubule. Mainly reabsorption occurs and begins here in a nephron. PCT is where reabsorption begins. It is located mainly in the cortex, as I said, and it is lined by brush border epithelium. And why it is called brush border? Because it has microvilli, as you can see in the ultrastructure form. It has mainly cuboidal epithelium because the cuboidal epithelium is arranged 3D structurally, like up and down and all. So you cannot mainly see that square form, but you see ideally this cuboidal columnar and all, it might be not like exact square or rectangle as you see imagine, but this is how ultrastructurally a cuboidal epithelium looks like. So PCT is lined by brush border cuboidal epithelium. Function of PCT is to reabsorb 65% of the filtrate, which contains mainly water, glucose, amino acids, and ions. And it also has sodium, potassium, ATPase pump that will help in the reabsorption, which is energy dependent reabsorption, and which is ATP dependent. The next we come to the loop of Henle, and you know loop of Henle has assisted vasa recta or the capillary loops. Okay, the capillary loops which are covering the loop of Henle are called vasa recta, which has artery as well as arterial or venous as well, because you can see the red color and the blue color. And so now the glomerulus, the urinary space, from there urine passes through the PCT, it gets absorbed water, glucose, amino acids, etc. with energy dependent sodium, potassium, ATPase help is there, and then the urine passes through the loop of Henle here like this. So the loop of Henle has two components, that is thin descending loop that is more permeable to water, to reabsorb water and concentrate, and then there is thick ascending limb which will reabsorb the ions and it is impermeable to water. Here it is impermeable to iron and it is permeable to water, and in thick ascending limb it is reabsorbing ions mainly. Okay, so when water is reabsorbed, the urine gets concentrated. So that is why in juxtamedullary nephrons where loop of Henle is too long, there is more concentration of the urine. Okay. So you will excrete concentrated urine according to the hormone and metabolism which is present in our body. So location, it is found mainly in the medulla inside loop of Henle, and it's critical for urine concentration. And then it also maintains the osmotic balance in our body and the fluid electrolyte balance. And it conserves water because it is concentrating the urine. Next, the part is distal convoluted tubule where fine tuning occurs. That is, the cells that are lining are having fewer microvilli compared to PCT. They also have microvilli, but it is fewer compared to the PCT. Okay, and they are lined by the cuboidal epithelium. And then they are also mainly located in the cortex of the kidney, and their function is mainly to reabsorb sodium and water only. And they secrete potassium as well as hydrogen ions under hormone control to do electrolytic balance, okay, acid base balance. And these sodium water reabsorption are mainly affected by aldosterone and antidiuretic hormone that is produced by the pituitary. So you should know other thing in DCT is the juxtaglomerular apparatus. Okay, juxtaglomerular apparatus is formed, as you can see here. There is glomerulus and then the PCT, loop of Henle, and the DCT that goes looping like this. So the DCT part which comes close to the glomerulus, there is the juxtaglomerular apparatus. In the juxtaglomerular apparatus, there are three components, that is macula densa that is formed by the DCT cells. Then there is granulosa cells that are modified smooth muscle cells that are located in the afferent arteriole, that is bringing blood to the kidney. Okay, the afferent arteriole has the JG cells that are modified smooth muscle cells. Then there is the extraglomerular mesangial cells. Mesangial cells are supporting cells inside the glomerulus. There are also extraglomerular, outside the glomerulus, there are some mesangial cells. So these three components, that is macula densa and extraglomerular mesangial cells and the JG cells, constitute the juxtaglomerular apparatus, and they are mainly responsible for synthesizing and storing and secreting renin, which is an enzyme which is important for renin angiotensin aldosterone mechanism. So JG cells mainly they secrete the renin that are present in the afferent arteriole. The macular densa, they are especially to identify and detect the sodium concentration. So when your BP decreases or sodium concentration decreases, JG cells are activated and renin is secreted. Extraglomerular mesangial cell supports the function of the JG cells and the afferent and the macula densa. So now coming to the collecting duct, that is the final part of the nephron, there is again water balance only. So the collecting duct, as I showed in the picture, it is mainly located in the medulla along with the loop of Henle, and they collect, one collecting duct collect from multiple nephrons, the urine. And cell features are they have distinct cell boundaries and they have selective permeability to, especially selective permeability to water. So if you want to absorb water more, that final water absorption occurs in the collecting tubule, and that is also modulated by ADH mainly. And then they drain into the papillary ducts that go drain the urine into the renal pelvis. Now coming to renal vasculature or the blood supply. As I had shown, the renal artery forms afferent arteriole and efferent arteriole that enters and exits the glomerulus. And then there is peritubular capillaries also that help in surround the cortical tubules for exchange and reabsorption. And then there is vasa recta that I showed, which is the loop which is accompanying the loop of Henle. They also help in fluid electrolyte balance. So see, if you are seeing the kidney in this, blood brings the waste through the renal artery and they go like this branch and then they form arcuate arteries and then they enter the, in the cortex there are glomerulus in which they branch and then they form the afferent arteriole that goes into the glomerulus, efferent arteriole that takes away the blood which is filtered and it goes loopingly around the loop of Henle and it forms vasa recta, and from the vasa recta the venous part takes off the blood which has been filtered out of the waste. Okay, and the filtered blood goes outside the kidney. Okay, so this is unfiltered blood that enters, and after going through the PCT DCT the urine is excreted, which is the waste is excreted. So the vein that goes out of the kidney has the filtered blood that goes into the circulation. Okay, so from here glomerulus the blood is filtered and the filtered waste products go through the urine like this. And then if you need essential water reabsorption, iron reabsorption, that also occurs along with this. This is the summary of the kidney functions. So in histology, we have to know that there are four basic morphological components in the kidney, that are glomerulus, tubules, interstitium, and the blood vessels. I will repeat once more. It is very important. There are four components of renal morphology, that is glomerulus, tubules, interstitium, and the blood vessels. So as you can see, in reality, the glomerulus is only this much and major part is formed by, this is the afferent arteriole, efferent arteriole, that is blood vessels, and then there is the tubules present. The component in between is the interstitium. So you have the glomerulus, the tubules, the interstitium, and the vasculature. Okay, here the afferent arteriole, efferent arteriole, and the blood supply and the vasa recta, they constitute the vasculature. So if you see ultrastructurally, this is actually how kidney looks like in ultrastructure. This is the glomerulus, especially in the cortex only you will see this. This is glomerulus, and you can see here they are surrounded by the tubules. This is the cut section of the tubules. So kidney, this is how histology, if you are seeing in a simplified way, there will be glomerulus. If you see juxtaglomerular cells, which is little rare to find in histology, if you can identify, then there will be glomerulus. There will be tubules, which has PCT, brush bordered, and DCT, which has low cuboidal epithelium. And then you have collecting ducts, and then there will be interstitium in between, and there is also vascular supply. So if you see histology, that is cut section, you will see the glomerulus. Then you can see the Bowman's capsule, and the white side, a white space outside the glomerulus is the urinary space. Then you can see PCT, which is much more pink color epithelium, which has simple cuboidal with microvilli. And then you have DCT, which has simple cuboidal epithelium, only microvilli is very less, so you cannot I appreciate it so much. And then there are also some vascular vessels also that you can identify little less in this slide, because capillaries you will identify, or vessels you will identify mainly by the presence of RBCs in it. If you get the main slide, you might identify it better. So there will be vascular supply, there is glomerulus, there is tubules, and then there is small between interstitium. If there is pink areas, if you can appreciate it is better. But when diseases are coming, the interstitium will proliferate more, so you will appreciate it more. So according to the four components, that is glomerulus, tubules, interstitium, and the blood vessels, the diseases are divided and pathology is studied accordingly. So glomerular disease comprise mainly of glomerulonephritis. Tubular disease that affect the tubules are mainly acute tubular necrosis. Interstitial diseases are pyelonephritis. Vascular diseases are nephrosclerosis, etc. These are the main most common diseases I am telling. And then you will come to cystic diseases of the kidney, and then finally you will study the renal tumors. So summarizing the key structures, nephron is the functional unit of kidney, and you have four main components, that is glomerulus, interstitium, tubules, and the vasculature. The function of the kidney is mainly to filter blood, reabsorb the essential water and ions, to maintain the electrolyte balance, and then concentrate urine effectively. Clinical relevance is according to the histology which is there. You will study how to diagnose the kidney diseases. So thank you for your patient listening. So I have made this presentation from the reference of mainly Robbins' Pathology of Disease and then Guyton and Hall of Physiology. So kindly do like, share, and subscribe my channel, and then comment your doubts or any topics which you want me to teach. Okay, thank you so much. Have a nice day.